Okay, so for meiosis, the steps that go in there are interphase, prophase 1, metaphase 1, anaphase 1, telophase 1, and then we go on to prophase 2, metaphase 2, anaphase 2, and telophase 2. At interphase, you start off diploid, where the cells are being replicated. At the end of telophase 2, you end up with 4 haploid daughter cells with unreplicated chromosomes. There is no interphase 2 because essentially nothing would happen. Alright, so let's go through this step by step. So at interphase, the chromosomes are random and they're being replicated. One from mom, one from dad. Now over in prophase 1 or early prophase 1, the nuclear membrane is starting to break down. Also, the chromosomes are condensed and they found each other to form, I believe at this point it's to form tetrads. And then at the end of prophase 1 or late prophase 1, that's when synapsing happens, so they cross over so that there's genetic variation. Now, also spindle fibers are forming and they're getting longer. And then we go over to metaphase 1, where all the chromosomes, the tetrads, line up on the metaphase plate. And then the spindle fibers attach at the centromeres of these chromosomes. And then at anaphase 1, the chromosomes get pulled apart by the spindle fibers, and then we go on to telophase 1, where and cytokinesis, where you now end up with 2 haploid daughter cells. So you started off diploid, and now you end up with 2 haploid daughter cells with replicated chromosomes. Now we go straight into prophase 1, and the only thing to note about prophase 1 is that the spindle fibers are beginning to form again, and now the spindle fibers in each of the 2 haploid daughter cells are going to they'll attach the centromeres of the chromosomes, and so now this is metaphase 2. So metaphase 2, all the chromosomes line up on the metaphase plate. The spindle fibers attach at the centromeres of these chromosomes, and now sister chromatids split apart. The thing to note about chromatids is that this only applies to chromosomes that have been replicated. So sister chromatids are now separating, which is anaphase 2, and then telophase 2 and cytokinesis. This is where now you end up with 4 rather, haploid daughter cells with unreplicated chromosomes. So at the beginning you had interphase, which was a diploid parent cell with and the chromosomes are replicated. So let me repeat that. Interphase is where the chromosomes are being replicated. This is a diploid cell, and this is just one cell. And then at the end of telophase 2, you ended up with 2 haploid daughter cells with the chromosomes that are replicated. Now at the end of telophase 1 rather, sorry. So interphase, now telophase 1, you ended up with 2 haploid daughter cells with replicated chromosomes. And then at the end of telophase 2, you ended up with 4 haploid daughter cells with unreplicated chromosomes. So that is meiosis in a nutshell. I hope this helps you guys out. Alright, see you guys later.